amino acid structure, you should know it has an amine group,
Yeah,
an
there's like
R
the
group.
C, then the
Mm.
carboxyl group, the NH2,
Yes, so that is
and
very important. And this R side chain decides the amino acid, what type of amino acid it is. It might have sulfur, it might have nitrogen, and so based upon that, amino acids are being named. So four different groups, you call it as carboxyl group, COOH, NH2 amino group, hydrogen, and a unique organic side chain, which is R group. And R group can be polar, non-polar, linear, ring form. So in 20 different amino acids, each of the amino acid has a 20 different kinds of R group. So some will be just CH3, some will be a ring shaped structure. So same way here also, there is condensation reaction, that is removal of water molecule. So, an hydrogen group from the amine and the OH group from the carboxyl atom, from the carboxyl group, they release the water molecule, resulting in the formation of what type of bond?
covalent.
What bond is formed here?
I want to say glycosidic linkage, but I know that's not the
That is
answer.
carbohydrate. Glycosidic is like carbohydrate. Ester is fat. What about this?
Is this something about polypeptides?
Yes, it's a peptide bond. So when it's formed between two amino acids, it's a peptide bond. When it is formed between many amino acids, it is a polypeptide. And so it is a polypeptide structure, right? And so this is the structure is important. So you should know which one is the N terminal, which one is the C terminal. The amino group is represented as N, and carboxyl group is represented as C. And the so all that labeling you should know. So there are two different types of amino acids. One is essential and non-essential. What are they?
I couldn't tell you.
Okay. So essential amino acids are something that you need to take in through your diet. And it is not
Oh,
synthesized.
and then the non-essential ones are the ones your body already creates or
Yes.
produces.
So that is
Okay.
directly broken down by your body, and that is non-essential amino acids. So you need not take them through your diet. And so we have this vegan diet nowadays becoming very famous, which is full of what is this plant-based protein. So that is vegan diet.
Mm-hmm.
Okay, coming to essential, non-essential amino acids, let's move ahead. Coming to protein structure, there are four different structures: primary, secondary, tertiary, and quaternary. Primary structure is what you saw now, like the amino group and the carboxyl group, condensation reaction, formation of a peptide bond or a polypeptide. That is the primary. So it's a chain
So
reaction.
just so
Mm,
just the polypeptide chain is
correct.
the
Yes.
primary structure.
Yes, absolutely right. So, what we were saying, so in this case, we say that proteins play a very important role in deciding the genetic characteristics or the amino acid sets. So I think you would have heard about gene expression, right?
Mm-hmm.
The genes in the DNA gets transcribed into mRNA. mRNA gets translated into a protein molecule. This is what is called as gene expression. How a gene gets expressed in the form of a protein, which represents a phenotype of any living organism, right? So the genetic code which we are talking about is made up of codons.
Yeah, those are groups of three amino acids. Oh,
Yes, so
three
which are
base
nothing but
pairs.
three base pairs. So triplet codon, you call it always, they occur in threes. So it's a
Yeah.
triplet codon. So genetic code is said to be degenerate. Why? Because you have 64 different codons, but there are only 20 amino acids. So certain amino acids have repeated codons. And so some amino acids are coded by multiple codons. However, you have mutations, changes happening, leading to diversity, all that is there. And so this is a property of a genetic codon, which is mentioned here. are okay. So polypeptides. So many peptide or many amino acids together forming, resulting or linked together by a peptide bond, you call it as a polypeptide chain. So single chain polypeptides are examples we can give enzymes and hormones. So these single chain which I am talking about is a primary structure. So I told you, no, straight line, just many
Mm-hmm.
amino acids making a peptide bond altogether. So,
Mm-hmm.
enzymes and hormones usually share this primary structure of a protein molecule, which is just a linear sequence of amino acid that makes up a protein. So, example, they have given lysozyme. Lysozyme is made up of 129 amino acid. So, you know, lysozymes is present in saliva and it is antibacterial property. Next is alpha
Mm-hmm.
neurotoxin, which is nothing but that is present in a snake venom. And it directly attacks the nervous system, and it is about 60 to 75 amino acid. Glucagon, so regulating blood sugar level. Myoglobin, myoglobin is an oxygen binding protein. Myoglobin is present where? Respiring tissues,
Um,
muscle tissues. In muscle tissues, you don't like, instead of hemoglobin, you have this, not instead of, you have myoglobin in muscles. In muscles, you have myoglobin. That's the oxygen binding protein, like a hemoglobin in the blood. In a muscle,
okay, okay.
so that is also a protein molecule which is made up of 153. So all these examples which I am giving have a specific primary structure, which is a linear sequence of amino acid. Just many amino acid formed together by a peptide bond. Is it
Okay,
fine?
do I need to know these examples?
At least two or three, whichever is easier for you.
Okay, I remember the lysozyme, the glucagon, and the
Myoglobin.
myoglobin.
Okay.
Also,
So
I want to
coming to
ask,
what am I?
do I need to like,
Neurotoxin. Yeah, please remember alpha neurotoxin also. Sometimes I think somewhere I don't remember, but snake venom, that I remember somewhere that question having.
oh, okay,
Mm,
okay. I have a different question though.
yeah, yeah. Tell me.
Um, do I need to know the different amino acids by heart?
Different. No, 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 not essential. No, not
Because
essential.
then, like, how am I supposed to know which part is the R group and which one codes for which amino
No, no,
acid?
that is not essential.
Because literally in the paper I did, there was a question about which
Mm.
part is the R group, and I got it
I'll
wrong.
tell you how to make it simple. You have sulfur containing amino acid. That is a common one asked. So remember sulfur containing methionine, cysteine. These are very specific. Okay. Sulfur containing amino acids. Homocysteine, taurine, taurine also. No. Just remember methionine and cysteine. So that is a very common example that. So this type of question, sulfur containing amino acid or a an amino acid with a sulfur group asked, and you're asked to identify what type of amino acid. At the time, you can you can choose either cysteine or methionine. Methionine is the start amino acid. AUG codon on codes for methionine. That's the starting one. Yeah, all these are new, or why? Why is this reaction?
Because I've never heard of that before.
Okay. So there is a start codon, right? Like for an for a process to start, that is this translation to start. There is a start codon AUG. Okay, AUG codes for amino acid methionine.
Uh-huh.
Okay, so like this, not all things is nowhere it is asked to remember, and I have not seen questions like that also. Remember certain things that are very different from the others or common, like as I told you, special cases like sulfur containing is a special case, so you can go in for cysteine, in methionine, and like that, choose specific ones to learn.
Okay. And like,
All the things is not possible also. 20 amino acid they will not ask, only in PGUG. There also we have not studied. Okay, yes.
and do I need to know
Hmm.
the the different R groups? Like what happens if it's hydrophobic, hydrophilic, polar, charged, acidic, or basic, if it contains the hydrogen ion
There.
acceptor?
No, 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 no. If you are able to, okay, but if you're not able to, don't take the strain. And because as far to my knowledge, maybe in your schools to make it little more harder, we can give such questions, but in examinations, I have not seen.
Yeah, like my teacher in an exam and like an internal test,
Yeah, that
she
is maybe to
she
make
put
your.
a question and I was just like, I don't even, because like I honestly felt like I
What did
didn't know
she
that there were
ask?
different types of art. Like
Mm,
she
mm.
asked, um,
Yes,
like
as I told you,
she
the
gave like
R
an amino
groups are
acid.
yes. So
So she
there are
was,
different types of amino acid. Like as you told me, some have hydrophobic ends, hydrophilic groups, all that only. No, saying polar, uncharged side chains,
yeah, exactly. And she
electrically
asked me to
side
like
chain.
tell her the effect of it. Like this is the R group. These are like all of the things. Is it hydrophobic, philic, polar, charged, acidic, basic, or what it is? And I actually need
Mm.
to know that?
See, not actually need to know that. I'll tell you how you can easily identify. For example, if you take amino acids with electrically charged side chains, and so if I show you some amino acid, mostly you need not know that, but okay.
Like how
See,
will I know what to look
it
for
is
in the
not essential. It is not in the syllabus. I'm telling you, to make it little more difficult, might be your teacher would have
uh-huh.
asked you. I think so. I'm not sure of. So if you see some structures like this, like this is how it looks like, right? And this is how all the side chains are. These are not, the amino acids is not given here. So if you want to identify which one have a positive charged, all ones which have NH2, right? You can see NH2 over here. Alanine. Can you see?
And glycine and isoleucine and leucine and
Lysine. There is lysine. So if you see here, all that can be considered as electrically charged side chains with positive groups.
because it's NH2.
NH2, yes, because it is NH2. Okay. On the
Okay,
other
so
hand,
NH2 means
if you
positive.
see, Otama,
So NH2 means it's positive, positively charged.
yes, NH2 positively charged. See here, you can see minus. Can you see here minus in the O? Aspartic
Where?
acid. Here.
Which one?
Aspartic acid. The arrow which I'm showing. You're not able to see my screen.
I am, but I'm trying to find it.
Aspartic acid here can you see? I'm just scrolling. Can you see here?
Yes, yes, there we go. Uh-huh.
So that there you have a O minus. Glutamic acid, you have a O minus. All these are
Uh-huh.
electrically charged side chain. So if it is so, you can choose negative. Like you can choose them to be negative side chains.
Okay, and then like the arginine right
Yes.
next to the glutamic acid, I think that's a plus,
What
right?
am I?
There's
What
like a
did you
little
say?
plus right next
Plus.
to the arginine.
Yeah, so these are all. So one which contains NH2, O minus, all these are considered to be electrically side chain amino acids with electrically charged side chains. If you come to polar uncharged side chains, let me show you one example. Polar uncharged side chain, this serine.
Mm-hmm.
Can you see this serine or serine, whatever it is? As,
Yeah,
so here you have an NH2 and double bond O. Can you see? But it's
yeah.
not free for it to bond with the other. It's already completed, this double bond O. Here and all
But
it is
all
see.
of them have double bond O.
Here.
Yeah, but that's what makes them charged. Like, so then what's the difference?
No. I'm talking. Here you cannot see a free O.
Oh yeah, because they're both bound to like both N
Yeah.
ones are
So
bound
they
to
yes, so
H.
they are completely occupied. So this is all
Okay,
chemistry
okay,
involved. Okay. So like this, you can remember something, but it is not essential that we need to memorize it. Nowhere in the syllabus there is memorize, that thing is not there. And maybe you can
okay,
remember few examples like what are hydrophobic, just two examples of each at least. Especially sulfur, I have seen. They have asked special case. Sulfur amino acids are very common and very specific. So they would give structures, or they would ask to identify what are sulfur containing amino acids, that I have seen. But I have not seen like hydrophobic, hydrophilic, and such questions are mostly rare, or I have not seen as of recent papers. I have not seen. Might be to make you
okay.
little into it. They have. Okay, the next
Yes,
one
yes.
is the effect of pH and temperature. So you would have heard about denaturation. So what is denaturation?
It's when the active site like just doesn't work anymore due to maybe high temperatures or differences in pH. The enzyme, the enzyme and the substrate just don't bind together anymore
Okay, why do they don't bind together?
because maybe the structure of the enzyme can change. So
Yes, that
like
is what I asked. Yes. So the structure of the protein, which example is an enzyme. So when there is something called as optimum, so optimum temperature, optimum pH, or whatsoever. So when that
mm-hmm,
goes beyond the optimum, the bonds become the hydrogen bonds that make up the protein molecules become very weak, and they start at breaking down. As a
breaking.
result, the proteins unfold and then lose their function. And so for
Okay.
humans, it's mostly 37 degrees Celsius, and pH is neutral pH. Above that, so for each enzyme and different protein molecules, the optimum temperature varies. It's not the same. So coming to the
Mm-hmm.
structures, which is important here, primary, secondary, and tertiary structures. So primary structures, as I told you, it's a linear sequence of amino acid held together by a peptide bond. So this is the structure of a protein, primary structure of a protein. And the amino acid sequence will determine the um, how the
Gene?
polypeptide chain will get folded. So this folding, which is happening due to the amino acid sequence, and will eventually result in the three-dimensional structure. As of now, what you're seeing is a just a sequence of a protein held together by a peptide bond, right? And
Uh-huh.
so this chain of sequence and even more folds leading to the three-dimensional structure, which we'll see in the next thing. Secondary structure comprises of two different figures or two
Oh my
different
God, I know these.
components.
Yeah, there's an
Hmm.
alpha helix, which is what's shown
Hmm.
there, and then there's the beta pleated sheets.
So alpha helix. So helix is the twist, like a DNA structure, right?
Mm-hmm.
And so this alpha helix, you can see them being formed between the hydrogen group of the amine, hydrogen atom of the amine group, and the oxygen atom of the
Carboxyl.
carboxyl group. Yes. So this is one amino acid and this is another amino acid. So since that bond is being repeated throughout, it happens to have a coil-like structure. That is alpha helix. So this is how you
Uh-huh.
write the explanation in examination for the extended response also. So alpha helix is the bond is formed by the hydrogen bond formed between the hydrogen of the amine group of one amino acid and the carboxyl group of another amino acid, which keeps on repeating. So this repeated pattern of hydrogen bonding allows the polypeptide chain to coil and give this alpha helical structure. This is how the response should be given. The next one, which is the beta pleated structure. So we know about the pleats, right? So it's like a, no, we don't say it as it is not folding, but it's pleats one over the other. So when you take,
Mm-hmm.
how does this beta pleated sheetings form is as these two chains run anti-parallel to each other. So you can see the hydrogen bond formed between in the adjacent strands. So between the adjacent strands, an hydrogen bond is formed, Same way by the hydrogen of amino group of one amino acid and the oxygen of carboxyl group of another amino acid. So this is what it gives a pleated nature or a pleated structure.
So they're formed by anti-parallel
Yes, the
amino acid
polypeptide chain.
polypeptide sequences, polypeptide chains.
Yes, polypeptide chain which run anti-parallel to each other, and the hydrogen bonds here are formed between adjacent strands. That's the difference. That's why there is no coiling.
Mm-hmm.
So this is the alpha helix, beta pleated primary structure and all is very easy. It is easy for us to explain. Shall I move forward?
Sorry, now you can.
Okay, so coming to the tertiary structure. So tertiary structure is, as I told you, all these are the sequence of the amino acid is making them. So as the amino acid bonds on, there are hydrogen bonds that is formed between one amino acid and another amino acid. So we know that each of the amino acid has their own structure. So when that bonding is tending to happen, there happens to be folding, pleating, and furthermore foldings. So with this furthermore folding will result in this tertiary structure. And here, why is it common instead of using alpha helix or beta pleated? Further folding is represented as tertiary structure. Why? Because it involves the formation of other bonds, including hydrogen bonds. So far, we were talking about just the hydrogen bonds formed between hydrogen and
Oxygen.
oxygen of each one. But here it involves other bond formation, which is ionic bond,
Disulfide bonds?
yes, disulfide bond hydrophobic interactions. And so this tertiary structure, since so many bonds are present, it is a very stable form of a protein. Like it is a more tensile and very strong kind of protein. So we already know hydrogen bonds are in other things. We saw that hydrogen bonds are formed between hydrogen of amino group and carboxyl group of another amino group. Here, the hydrogen bonds are formed between the polar R groups.
So like, for example, if an amino acid isn't polar, then it won't have a tertiary structure.
One amino acid, you have so many one protein is made up of 60, 70, 80. How come there will be no polar itself?
Okay, but but since there are 20 amino acids, and
Even
what?
amino acids, it will be a combination of hydrophilic, non-polar, hydrophobic, polar, everything only.
So it
Just
has
polar,
to be a combination. It
it
can't just
will
be
be.
maybe like the bunch that are just not
It
polar and
will
then just
not,
make
it
a
cannot
chain.
be, no, if only when they're opposite charged molecules, only when they are electrically charged, or only when they're opposite in nature, they will be able to form a bond by sharing electrons. If everything is similar, then where will the bond formation happen?
Oh, right.
Correct. So there must be, for any bond formation to happen, all that sharing of electron and transfer of electron, losing of electron, gaining of electron, everything. That is what is bond formation. So in that
Correct.
case, that has to happen. So in that case, there should be a combination of all these things. So what they're trying to say here is the extensive folding. Now until now we were talking about this amino acid just bonding. That's why there is a folding or a pleating. But here, if the hydrogen bond is just formed between them two, then that extensive folding cannot happen. And if there are many amino acids together, to bring them together, you need such things to happen. For example, it's made up of 300, 400 amino acids. To just pile them up together, you need extensive folding. When will that happen? When one distant from first amino acid to 100 amino acid, when there is a bond is formed. So that can happen between this hydrogen bonds we are talking about, right?
So it's
And
just the further, so tertiary structure is basically just the further folding.
yes, further folding to bring them up together. Yes, correct.
Further
So
folding of,
hydrogen bonds. So this is helpful in maintaining the functional integrity and providing them the correct structure. Ionic bonds, we know ionic bonds are usually formed between opposite charged ions, right? And so here also the same way, the charged R groups. So if it is a positive R group, they just form an ionic bond with a negative R group, and they eventually form an ionic bond. So this also helps in maintaining the integrity of the protein molecule. Disulfide bond. Already we know that sulfur containing amino acids are present. So in case of sulfur containing amino acid, that is cysteine, so when both the sulfur comes together, then you call that as disulfide covalent bonds. And so they also play a very important role in maintaining the 3D structure of the protein molecule. Hydrophobic interactions. So hydrophobic interactions happens between what type of amino acid?
What?
Hydrophobic interactions and happen between what type of amino acids?
Hydrophobic.
Yes. Polar or non-polar?
I think hydrophilic is polar, so hydrophobic
Yes,
is
hydrophobic is
non-polar.
non-polar amino acid. So hydrophobic interactions happens between non-polar amino acid. And so when water or we know that non-polar amino acid cannot combine or cannot react with water, right? And so in that case, what happens is all the non-polar amino acids come together as clusters inside the protein so that they don't come in contact with the water molecules that is outside. That is called as
Mm-hmm.
hydrophobic interactions or hydrophobic bonding. You get it?
Yeah. Can you
So when
repeat the thing
these
for hydrogen bonding again? I
hydrogen
couldn't.
bonding is formed in the R group of the polar amino acids between the polar R groups in the tertiary structure of a protein, and it is able to maintain the 3D structure by making all the distance amino acid come together. Uh,
Oh, this is what forms a globular protein.
yes,
So the tertiary structure is the globular protein.
yeah, it is the globular tertiary structure is globular protein is quaternary. See, I am telling you, all the amino acid exist in all four forms. It starts with primary, moves to secondary, tertiary, and
Then
quaternary.
quaternary.
Yes, so it is formed like that only. It just cannot, but it exists as a quaternary structure. You're getting it. It cannot remain as a, because it's a large protein molecule. It's not just a simple protein, not just one amino acid or two amino acid, right? And so, the globular protein is a spherical shape one, and it is having a hydrophobic interior and a polar exterior, right? And so, in
Uh-huh.
that case, is you can say that it is a tertiary structure of a globular protein is having this hydrophobic interactions where they have all their hydrophobic regions in the interior with the polar groups outside, polar amino acid outside. Like that, we can explain in terms of tertiary structure. You're
Mm-hmm.
understanding my point, what I'm saying?
Yeah.
When two or more globular proteins come together, it becomes a quaternary structure. It's a multimeric unit. You get it?
Uh-huh. Yeah.
You're understanding? That's what. So you can explain the globular protein in all terms.
Yeah. So it starts off with the polypeptide chain, which is the long chain of amino acids, then the alpha helixes and the beta pleated sheets, which
Hmm.
are then formed by, because the alpha helix sheets are formed between descending
Hmm.
H groups and the oxygen from the
Hmm.
carboxyl group. Then it forms the coiled sheets or anti-parallel polypeptide chains, and the hydrogen bonds are formed adjacent to one another. Again, the H from the amine group and then the O from the carboxyl group. And the tertiary structure further is just further foldings of the chains via different bonds, which are hydrogen bonds between the R groups of polar amino acids, ionic bonds between opposite charged ions, disulfide
Hmm.
covalent bonds, which are cysteine amino acid residues, and then the hydrophobic interactions between non-polar amino acids, and then they just bring a like amino acids together to form globular proteins.
Yes, right.
Okay.
Okay, so what we were so these are the examples that you can see here. See, hydrogen bonds between distant amino acid, hydrophobic regions, just all the non-polar amino acid towards the interior, disulfide bond between two sulfur groups, and ionic bond between opposite charged ions, right? So this is how this structure is made. So here you can just see how a spherical nature is formed, right? So you can explain proteins in terms of all these things. And so what is the effect of polar and non-polar amino acid on the tertiary structure of a protein? And how do they influence the tertiary structure? Now, we just now had a thing, right? How are they making how is this polar and non-polar amino acid determining the tertiary structure of a protein?
It just determines
Now,
the
just
shape
now
of
we
it.
were discussing, right? And based on their charges, based on their polarities, they interact with the water molecules. So polar means they can very well
They're
interact with the hot.
hydrophilic.
So
So then the
they're
hydrophilic amino acids
correct.
would be on like the outside, and
Yes, so
then
and hydrophobic
hydrophobic.
towards the inside. So this is how they form the tertiary structure. If there is no polarity or the interaction with the water molecule doesn't differ, then this type of structure is not possible. So we take the example of myoglobin, as I told you, that is present in the muscle tissues, and even hemoglobin. Both are examples of globular protein, which we discussed now, right? Whether it is myoglobin or hemoglobin, and both are examples of globular protein which has hydrophilic ends exposed outside and hydrophobic ends towards the interior. You would have also heard about integral proteins. Where did you hear about integral proteins?
Oh my God. In the phospholipid bilayer, they're like embedded
Correct.
in.
Yes. So one example is rhodopsin. Rhodopsin is a protein found in the cell membrane of the retinal cells, retina. All right. And they are responsible for observing light. So this is one of the integral protein that has their hydrophobic regions. So this is also an example of the tertiary protein which we are talking about. This is how the structure, three-dimensional structure looks. Next, we are moving on to quaternary structure of the protein and form and function in globular and fibrous protein. So here only we are going to talk about the form and function of this quaternary structure in globular and fibrous proteins. So again, it is furthermore arrangement. Tertiary structure itself is a congested folding arrangement. And so furthermore arrangement and interactions and of two or more polypeptide. As I told you, two globular proteins together. This is also a polypeptide. This is also a polypeptide. So when both of them come together, you call that as that interaction, that arrangement. You call that as quaternary structure. Is it okay?
But aren't the beta-plated sheets made of two chains? Two
Yes.
anti-parallel
I'll
chains?
tell you. So, for example, hemoglobin, no, it is not two polypeptides. They are forming a polypeptide.
No, but like you said, na, that like the two of them that go the other way around, and then with the hydrogen and
That is
oxygen
not two
bonding, they
amino
make like a.
acids. See here, it is one strand. If you take this structure, it is just one strand where the amino acids are arranged.
Yeah, for the beta-plated sheets.
Beta plated sheets, they are folded adjacent strands.
Oh, okay.
They are not different polypeptides. This is one polypeptide only. You
Uh-huh,
got
but it's
it?
folded in half.
Yes, they are just holding. All this is, it's a bigger molecule. It has to accommodate. Hemoglobin is present everywhere. As we are seeing, it's not that big molecule, right? It's a, it's
Mm-hmm.
a, it's a protein molecule. So in that case, it has to flow through your body. So how does it happen? It happens through these rearrangements and foldings and whatever we are talking about, formation of bonds. So if you take hemoglobin in a quaternary structure, it is made up of 4 polypeptides, polypeptide units. 2 alpha, 2 beta. Can you see here? Alpha 1,
Okay,
alpha
so
2,
there's
beta
2
1,
alpha
beta 2.
helixes and 2 beta plated sheets merged together.
Yes. Merged, we cannot say. They are forming interactions, bondings only, everything.
Okay, so they have interactions. Wait, so does that mean there are 4 separate globular proteins? So one alpha helix makes one globular protein, and one beta plated sheet makes a different globular protein?
What did you say?
So, like, does one alpha helix make
Hmm.
one globular protein? Makes
Yes.
one.
One. One protein, one polypeptide.
Okay, so then okay, so one alpha helix will
Hmm.
make one tertiary structure.
One alpha helix will make one tertiary structure. Yeah,
Or
okay,
is it
yes. No.
alpha
Correct only.
plus
No.
beta?
No. It's not alpha
Just
plus beta.
alpha.
See here, if you see the tertiary structure here, which is shown for rhodopsin, is
So
these are all
there's a bunch of alpha helixes.
yeah, alpha helixes, correct.
Okay, so you can't mix an alpha and a beta, for example. You can't mix them and make a tertiary structure.
That depends on how they get arranged.
Oh my god.
Now we need not worry about all these things. You just remember this.
No, because I'm trying to figure out how you make this heme group. So like,
He see it's this is a quaternary structure which we are talking about. So it has 4 individual polypeptide chain. That should be our objective. Our objective
oh,
is
okay. So four separate
yeah,
polypeptide sequences.
polypeptide chain.
Just
Yes.
2 of them coincidentally make the alpha helix, and 2 of
Yeah.
them coincidentally make the sheets. That's
Yes.
it.
Yeah. Yes. Correct.
That's it.
Yeah. That's it. Nothing else.
And
If 2
then,
alpha and 2 alpha, which is alpha 1 and alpha 2 and beta 1 and beta 2 come together, and there is an iron molecule where you can see them, the heme group attached, that is a hemoglobin. That's it.
okay, so then where's the tertiary structure?
Where do you
How
want
how
it?
do you just you've just taken 4 secondary structures, put
So
them
how will this?
together,
Okay.
and now you just made
Tertiary
a
structure.
quaternary structure?
What is how does tertiary structure get formed?
The further folding of the chains.
So
Chains,
here it is.
plural.
Here is it necessary for it to further fold? You need 2 alpha, 2 beta to make that one multimeric unit, right? This does, you understand now. Here the tertiary structure is not essential.
Yeah,
Not
but how?
everything has to exist in a quaternary form. Not everything has to exist in a secondary form. Not everything has to exist in tertiary form.
But you said everything exists in every form.
Depending upon the nature of the protein molecule, depending upon the nature of the specification, it is needed.
Also, it depends on the sequence.
Depends on the sequence, depends on where it has to be placed, what function it has to perform. For example, here now we talked about rhodopsin. Rhodopsin in the retinal cell. Rhodopsin doesn't exist as multimeric unit. It has to exist as a tertiary form in the retinal cell, because only in that structure it can absorb light. The function is so.
Okay. So, because here it does not need to be further folded, which is why they just take the structures and put them together with heme
If
groups
there
in
is
between.
some other protein in like if there is an alpha helix protein which need not or doesn't have the ability to combine with the iron, then it will go to formation of some different protein. This alpha has the ability, the sequence of the amino acid is in such a way that it has a binding affinity for the heme group. So
Right,
it
because of the
combines
specialized sequence of amino acids or the specific sequence
everything
of the
is dependent
polypeptide
on,
chain.
correct. Everything depends upon the sequence of the amino acid. That's why in the beginning itself, I told the sequence of the amino acid will determine the arrangement and the folding of the, or the further folding and representation of a protein molecule.
Okay, I'm gonna need you to repeat that sentence again. Wait, the what?
The sequence of the protein determines,
The sequence of the protein.
sorry, the sequence of the amino acid. I'm sorry.
Okay,
The
the
sequence
sequence
of
of
the
the amino
amino
acid chain.
acid, the sequence of the amino acid, just amino acid. I even had that same sentence over here when I mentioned it. See, the unique sequence of the amino acid determines how the polypeptide chain will fold. That may lead to the three-dimensional structure of a protein. The sequence, the unique sequence of the amino acid will determine
Oh,
the
right, yeah, because the
arrangement.
tertiary structure depends on the R groups, the opposite charged ions, the
Yeah,
cysteine
that's what
amino
I told
acid
in the beginning
residues.
itself, right? So only the sequence will determine what type of amino acid, what type of a protein will it be formed, will it can accommodate. As I told you, there may be sometimes mutation. So if there is mutation in the hemoglobin in the alpha helix, it may not have the affinity to bind with the heme protein, and it may not form a hemoglobin. So there are possibilities. So this unique sequence of the amino acid will determine what type of a protein will, what type structure of a protein molecule it should form depending upon the polypeptide folding and arrangement.
Oh, okay. Okay, now I remember because the tertiary structure relies on R groups, on the R groups and the oppositely
Not only
charged
R
ions
groups,
and stuff.
tertiary structure, hydrophilic, hydrophobic. You have so many different bondings that is formed, right? We even talked about how that hydrophobic arrangement, polar amino acid makes up
Yeah,
the
and all of that
outside.
depends on the sequence of the amino acids. So some
Yeah.
of them
So
can't
if
have
they,
tertiary structures.
so only the amino acid sequence will determine what type of a structure it should be formed. And also it depends upon the location and its function. That is also there. Right?
Okay.
So our major aim in terms of assessment is just explain how an alpha helix is formed, how a beta sheet is formed, because they don't ask you whether this amino acid is capable of forming a tertiary structure. This amino acid is capable of forming a quaternary. No. Explain the quaternary structure of a hemoglobin molecule. So in that case, we say that hemoglobin is what is it? And it has it is made up of 4 individual polypeptide chains, 2 alpha chains, 2 beta chains. And you also consider them to be a conjugated protein because it has a non-protein component, which is a heme that has affinity towards ants. And heme is nothing but iron. I think you know that. So iron is present in the center. And what is the responsibility of hemoglobin? What does it do?
Oxygen
Where
absorption.
does oxygen bind to?
Yeah, sorry, it doesn't absorb. It binds to oxygen, which is then carried around the body on the little red blood cells.
No, either heme group or globin. Which binds to the oxygen?
Heme.
Globin doesn't bind with the oxygen.
So
Heme only.
heme. I was
Yes.
right.
So heme plus oxygen. So it is carried as hemoxyglobin in the blood, and it is supplied, delivered.
Oxy.
The oxygen is supplied wherever it is required. So so each of the units which we are here, alpha and beta, is held together by non-covalent bonds, and certain interactions see some conformational changes that is happening in the structure, because as it has to undergo change in order to enable the binding of the heme with the oxygen. That's how they carry out their function. This is what you need to explain in terms of when hemoglobin is asked to explain in terms of quaternary structure. When you give these type of explanation, it is sufficient.
Okay.
Okay, next you have conjugated and non-conjugated protein. Conjugated protein, just now I explained. When it is present along with the non-protein substance, you call it as a conjugated protein. But non-conjugated protein is you don't have any other substances by it's all completely a polypeptide subunit like insulin, collagen, and all those are. So in conjugated protein, you can have a metal, you can have any other carbohydrate, for example, glycoprotein. In all those are conjugated protein. In enzymes, so enzymes are right. So all those can be considered as some kind of conjugated proteins. And you know the functions of enzymes. The enzymes are catalytic in nature, and
They're
because of these.
biological catalysts that speed up the rate of of a chemical reaction.
Correct. And this is about conjugated and non-conjugated protein. The last one is globular and fibrous protein. So as I explained to you, globular is spherical, right?
Yeah,
Soluble in water and
because
mostly
of the
globular.
hydrophilic exterior.
Correct. Yes, hydrophilic and hydrophobic interior. And they play an important role as enzymes, transporters, regulators. Insulin is the best example with two polypeptide chain. And so here you have an alpha and here you have a beta, but they are arranged in three-dimensional shape. Here you have all the bonds which we talked about in the three-dimensional structure. What are the three-dimensional structure we talked about? Hydrophobic interactions, disulfide bonds. So again, this is
Bitter
determined.
bonding and stuff.
Why did this did not become as a quaternary structure if you ask? Because there are already other interactions that we are talking about. There in quaternary structure, it is non-covalent bonds. This sequence of amino acid has determined this structure.
Mm-hmm.
You understood at least to some, now at least you're coming to some conclusion.
Yeah, now I know I won't make the mistake because I actually know something. I
Oh,
didn't bother learning it before. It didn't make any sense.
hmm? What am I? You didn't read it and come, huh?
Yeah, no, I didn't bother learning it before because I couldn't understand it. So
Okay, I think now you read it once again, you will be able to understand it a little in
yeah.
better. Just go through something so you will understand now better. So the sequence of amino acids. So here there might be some sulfur containing amino acid which has enabled them to form a disulfide bond,
Mm-hmm.
right? There would have been polar amino acids arranged in an appropriate, which would have enabled the different kinds of bonding that is required for a three-dimensional shape. That's the reason insulin is a globular protein and it has a three-dimensional structure. So we know about insulin, which we have discussed in the last classes also. It regulates
Mm-hmm.
the amount of blood glucose. So when your blood glucose level is high, insulin is secreted by the pancreas. So insulin binds to the receptors that is present, and that is how they initiate the activity of reducing the blood glucose level, and right. And this is about chemical signaling. So example of globular protein, if you need to explain, you can take insulin, which is made up of two polypeptide chains. One is alpha, another one is beta, because they have disulfide hydrophobic interactions. Coming to fibrous proteins, so they are not like globular proteins. They're much more simpler, very long fibers in nature, and they are insoluble in water. But they are majorly their function is to they are not like the globular proteins, which are acting as enzymes or transporters and regulators. They are mostly involved in strength and giving stability to the cell or whichever they are made up of. For example, your
Mm-hmm.
skin, your tendons, muscles, your bone, all this. Collagen is the best example. So, like how you explained insulin for globular protein, you can use collagen over here. So, collagen is a connective tissue in animals, and it is present in bone, skin, and cartilage. So, here you have three polypeptide chain, all are helix, three different colors you can see, right? And so, not very many colors you can see. So, the three polypeptide chains, which is again helical only, no beta pleated sheet here. And so it is long, thin, and it gives an elongated fiber nature like structure, the sequence of the amino acid. And so have you heard about van der Waals forces in your chemistry?
Yeah, in chemistry.
What is it?
Um,
Hmm.
aren't like there are three different forces joined together are called van der Waals forces, and I think they're relatively weak. Because hydrogen bonding, which is the strongest, is not part of them.
We don't want chemistry here. Here you can see this forces and all over there in this collagen, which we are talking about. And it's a triple helical structure which has van der Waals, which is held together by hydrogen bonds and van der Waals forces. As you told me, it is a weak intermolecular force. And it is very strong, flexible. So you can have some characteristics. That's about
And this
collagen.
is a quaternary structure.
Is this a quaternary structure? Do you think so? This is a quaternary structure?
I want to say yes because there are
Why?
multiple chains.
Because they are
There are 3 alpha helixes, so I want to say it's a quaternary structure.
pi. If there are 3 alpha helix, then it will be quaternary. Why must it be tertiary?
Oh no, wait.
Huh.
No, wait, let me think.
Hmm.
Okay, it could be tertiary if there are the different types of bonds in between.
Okay.
But it could also be quaternary because each alpha helix makes a globular protein, and then this could be a big fat like thing of globular proteins because the hemoglobin is a quaternary structure.
The quaternary structure. Okay.
So I was right.
Yeah, you are right. So there comes the end of protein.